Let's go. It's a great pleasure to me introduce you, Dr. Claudia Alvarez. Claudia is an excellent young researcher, and we talk about a very interesting topic. Claudia, thank you so much for accepting my invitation, and please go ahead.
First, thank you for the invitation, and it's a great pleasure to present my work here. And let me share my screen. And what I want to show today is a work that is about how protein structure changes. So the title is a little bit unusual in that what we are proposing is that the evolution of fold structure is based a lot in reuse, and not much is completely new. Let me see, I think I can't. Okay. So what we do with proteins is that we try to read the information that we have stored in the sequence of the proteins and in the structure of the proteins, very similar to how we would read a historical record. So here I'm showing you an example of a true historical record. And one of the things that we can learn from this is that something that we are studying, an object of study, will have a relative age of appearance in the world. So for example, here, this Sumerian clay tablet may have a relative age, but we can infer things just by looking at this object. So for example, we can say that this writing already existed by the time this clay tablet originated. So this is similar to what we do with proteins. Some proteins and some other macromolecules and biological molecules will have a history recorded that can take us back to the last universal common ancestor. So these molecules are very special, and we like to study them because they carry a lot of information. So these molecules that can tell us about the history of life are sometimes called molecular fossils. And the best molecular fossils are going to be the ones that are universally represented in the tree of life, and that have a historical record that can lead us back, that can be traced back to the last universal common ancestor. But there are also other types of molecular fossils that probably will not take us that back in time, but still are going to be very important to understand our past. So we are actually interested in understanding not only the last universal common ancestor, but also the events that occurred before this population existed. So if we really want to understand things that happened before the ancestor of all living organisms, we need to use some information that is going to be given by molecules that are called deep paralogs. Deep paralogs are duplication events that are going to tell us about things that occurred before the duplication. So if something exists and we make a copy, we can infer that the copy was made before the two molecules are present. So that's what is represented here. Oh, I see a message. Let me see. Oh, sorry. Okay, so if we have a duplication event like in here, we can infer that there was an ancestor before the duplication event. And what I want to understand is duplication event in protein domains or in protein structures. So what is a protein domain? It's an evolutionary unit, and it's also a structural unit sometimes. So in the image on the right, we can see a protein that has 3 independent units, one in blue, in white, and in green. And these units have their own history. The reason why we see these in 3 dimensions is because the polymers, the proteins are polymers that interact, that form interactions between them. So we will have, when these interactions are formed, the formation of a 3-dimensional structure. These structures are marginally stable. That means that when we put some types of stress on the protein will unfold. And this can be changes in temperature or in pH or in concentration. So these structures are not always very stable. So these structures are actually the base of many, or I could even say most of the functions of life. So proteins carry out at least partially all the functions of life. And we need them to be in their 3-dimensional structure to work. But not all combinations of amino acids will form a stable protein structure. So the protein folding problem can be divided in many different parts, but here I'm listing 3. So one of them is that not all combinations of amino acids will form a stable structure. The other is that we cannot, there are no enough resources in the universe to explore all the different combinations that can be formed with the 20 coding amino acids. And the last one is that once we have a protein that has a structure, it doesn't mean that we will have a diversity of structures coming out of this one single protein structure, because interconversion between structures is at least not as frequent. So then how do we explain the diversity of protein structures that work in our bodies to make all the functions that we know they do? And if we analyze the sequence and structure of the proteins, as I said, we can understand their past. And one of the first things that we learn from the analysis of sequence and structure is that many of these structures are connected. And what I mean is that if we compare different proteins, we will going to see repetition or reuse of some secondary structural elements or some shapes. And we are going to see also reuse or repetition of some small fragments of sequence. So this at least suggests that some part of this exploration of the universe of possibilities has been through the reuse of sequences. And this makes sense in biology because we know that there can be gene duplications, and then when the new generations come, they will use the gene duplications as a starting material to make new things. So using all of this information, what I have been trying to do is to infer relationships between these protein structures or domains and try to see if we can draw a tree of relationships between the different structures. So first, if we want to understand what's happening in a mechanistical way, one of the things we would want to do is to understand the cases that are easy to understand. So the best known case of protein structure evolution is circular permutation. So in circular permutation, we will see a very similar three-dimensional structure. So the two proteins that I'm showing you here are circularly permuted. And what it means is that the secondary structural elements, which are here shown as a cartoon representation, are going to be rearranged in a specific way. So the structure on the left has the two secondary structural elements in red on the first part of the protein, and the structure on the right has the same two secondary structural elements on the end, the C-terminus of the protein. And that means that in the sequence, in the linear space, the secondary structural elements have been rearranged. But if we only look at the three-dimensional structure that is represented here, we will see that they look very similar, almost identical. So it would look like having two starting points that are the secondary structural elements and just basically joining them together in two different ways. So in the first way, we would join first the red and then the blue. And in the second case, we would join first the blue and then the red. And this is exactly what happens when we do protein engineering, when we try to design a protein structure with a specific configuration. But in evolution, this is not exactly what happens. So even if it looks like this is the explanation, when these proteins evolve, this is not how the secondary structural elements get rearranged. So to show what really happens, we have to understand how usually genes diverge. So one of the things that happens is that there can be, as I mentioned previously, a gene duplication. So usually a gene duplication, and I think I have, no. Usually a gene duplication will generate a repetition of the structure. But only sometimes these duplications will make the protein structure to interact in a way that is not exactly a repetition of the original, the ancestor. So in this example here, we have the gene for the fold 1 that has been duplicated, and we can see here that the duplicate in sequence has the same order as the duplicate as the ancestor. So we see 2 reds followed by 2 blues, then 2 reds followed by 2 blues. But the new generation of proteins will do the structure radically different from the ancestors. So even if it looks very similar, the difference is in the hydrogen bonds. So the hydrogen bonds are formed between elements in the middle of the protein and elements in the termini of the protein. This is observed, so this is an experimentally determined structure. So we know that sometimes when a gene duplication occurs, this is what we will get. So now what happens in evolution is that sometimes after this new form of structure folding occurs, and there is divergence of the sequences, we will get a loss of the termini of the protein. And when this happens, and we compare only the ancestor to the derived state, we will see that there has been a rearrangement of the secondary structural elements. But we know that this rearrangement occurred through a series of very well-known steps. So here are the two different views of the circular permutation phenomenon. So on the left, we would have the mixing and matching hypothesis, and on the right, we will see the creative destruction hypothesis. And we would like to study this second case, the case in which through small steps in evolution, we will get the formation of new structures. So beyond circular permutation, which is a very well-known phenomenon, we wanted to study this in a larger set. So this is what we did. And this schema has the idea of what we are going to be looking for. So it looks a little bit complicated, but basically what we wanted to do is take many proteins that we know that are very ancient, that can be traced back to the last universal common ancestor, and then we want to gather all the information from the diversity in the tree of life and represent it in the form of protein profile, which is just a representation of this diversity, and then form groups. And these groups will inform us whether or not there has been a fold evolution event. And the examples that we took are the same examples I've been using for representations. And these are small protein domains that are called small beta barrels. And these small structures are very simple, are made of basically only beta strands that are fold together. And they are present in many proteins of the translation system. So that includes the ribosome itself, but also the amino acid tRNA synthetases, which are the proteins that charge the amino acid to the tRNA, and also initiation and elongation factors.
Okay.
Could you mute your microphones? Thank you.
So what we got after we did the search, as I explained before, so we took the 3 types of small beta barrels that are shown here that I didn't present. So there are 3 types of the most frequent beta barrels, SH3, OB, and CLB. The names don't matter much, but these 3 groups are essentially different in the linear connectivity of the secondary structural elements. So when we do a search of the similarities in sequence, we see many cases where there is similarity in sequence, even if the structure we know is not the same. So these cases are the key to unlock the mechanism of fold evolution. So everything that is in these yellow squares are cases in which the information has been stored. So here I made a representation just using the same type of fragment analysis as we would do in the circular permutation, just to show how the 2 types of folds, SH3 and OB, which are very different in the linear connectivity, but very similar in 3D, would look if we just had the pieces. So if we had a collection of pieces to make proteins from, we would say, well, let's just take this beta sheet and then this funny looking element and just 1 strand, and with that we can form an OB. But then if we change this last element for the blue one, we will form an SH3. And the differences will also be in the order in which we connect one to the other, one element to the other. But we already know that this in evolution probably is not how it happens. This is how our brains work, but not necessarily how evolution will work. And the other key that we saw is that the similarities in sequence are telling us that the connections between them go in a very deep way, even more than just the similarities of structure. And what I mean by that is that here we have the elements that are homologous in sequence, or that share sequence in the same color. And we can see that there is one strand that looks the same in the structure, but for one of the pieces, it changes the conformation. So with this piece of the puzzle, we can now put all the pieces together and understand how these two are related in the creative destruction model. So we started by just drawing the diagram of a normal duplication event. So that would be the schema on the left. So if we repeat the SRH3 topology 2 times, we will have something like this. So you can see that it's basically, I just took it and copy pasted it. But through divergence of the sequence and of the secondary structural elements, we can get from the schema on the left to the one on the right, where some secondary structural elements are going to be preserved, but some others will be lost. And in the end, we can go from one to the other just by loss of some elements and rearrangement in the 3-dimensional structure. So we showed these in sequence, and we wanted to know if our hypothesis could be tested using AlphaFold. So AlphaFold is an algorithm that allows you to input a sequence that you, well, any sequence, a biological sequence or a sequence that you design. And what the algorithm will do is, based on a training, it will predict what 3-dimensional structure it would make in nature. So what we did is, just using the information that we have in sequence for homology, we told it to fold 2 SH3 domains, and it created an OB fold. So it created a new domain from just the information of the ancestors, which is different. The same if we take now this newly formed OB and we duplicate it, but just remove part of the information here, the strand 3 and here the strand 5, and we told AlphaFold to fold this, and this creates a new protein structure that is now much more complex. So 2 things I want to highlight from this. I showed you in detail the story of the SH3-OB relationship, and I didn't go in detail on the OB-CLB relationship, but the story of the OB-CLB, the one on the right, is not only the story of creative destruction, but it's also showing that this mechanism allows nature to put more complexity in protein structures. So it may not be always the case, but this is a mechanism which generates not only diversity, but increased complexity. So this is the conclusion for our model on creative destruction. And what I would say is that once we have a protein folding domain, then through divergence and very simple mechanisms, many folds can emerge. So creative destruction acts on the level of domain, depends on fold plasticity, which is this ability of structures of form new hydrogen bonds, and resolves these similarities between disparate protein structures in a mechanism that is biologically plausible. And what it suggests, and it's in agreement what has been observed, is that the universe of protein structures is much better, it's more like a network than as a simple tree. So with that, I want to thank everyone that was involved in this work and all the discussions that we had, with Professor Lauren Williams, Anton Petrov, and Rohan, Peter, Eric Smith, and Jessica Bauman.